Students, today we will discuss about the topic 4.3 and 4.4 in this video, and we will go through KPI by KPI. So first KPI 4.3.2, state the overall equation for photosynthesis and track the fate of each atom. That's very important, like what's the fate of each atom. So we need to go over the overall equation, and we know that the overall equation is carbon dioxide plus water gives you glucose plus oxygen, where oxygen is lost as a waste product. So this is the equation, we know that, but they have given us the question that the complete balanced symbol equation for photosynthesis. So we know two things here. First of all, symbol equation. So carbon dioxide is CO2, water is H2O, glucose is C6H12O6, and oxygen is O2. One of the most important thing how to balance it is that you can put 6 with every chemical but not with glucose. That's it. So this is your balanced equation. From here now you can easily see that the answer is D. That's 6CO2 plus 6H2O gives us C6H12O6 plus CO2. So in this case you should learn the chemical equation as well as the world equation for this one. Question number 2. The O2 released during the photosynthesis comes from which of the following molecules? So you should remember O2 is an oxidized molecule which is coming from the oxidation of the water. That H2O is basically directly converting into O2 and results in the loss of the electrons as well. That's called as oxidation. So when H2O gives you O2, so this is basically oxidation process, and we should know also that loss of electrons is happening over here. Now, so you can easily see that the answer is C. Moving to the next question. So in this way we learned about the fate of each. So if water is oxidizing into O2, it means that the carbon dioxide is being reduced into C6H12O6. So you can also say that CO2 is basically the one which is converted into C6H12O2, and in this case when you can see H2 is added over here, so this process is called as reduction. So this is what we have to know. This is called as a reduction process. Moving to the next KPI, describe using the diagram preview of the two stages of the photosynthesis. So first of all, you should know that what are the two stages. So light dependent, and the second is light independent. Light independent also have alternative names. That's, for example, Kelvin cycle. So Kelvin cycle or dark reaction. So these are basically the names of the light independent reaction. It doesn't mean that it takes place in the dark, but it is the one that takes place in the absence of oxygen. So it's not necessarily depending on the light. Now there is a diagram given over here. In this diagram you can easily see that when number one, when the oxygen is produced from water, that's basically the light reaction, and in this reaction what we can also see that O2 is lost. That was also question number 2. Okay, and this process is taking place in these structures. The disc-like shapes are called as thylakoids, and the stack of the thylakoid is called grana, and inside the thylakoid we have chloroplast that contains a pigment chlorophyll, and that's basically responsible for the reflection of the light. My apologies, that's basically responsible for the absorption of light. Now the two products that you can see, the arrows which are going out, so ATP and NADPH, these are the two products of the light reaction. While on number 2 you can see that this is taking place in the stroma of the chloroplast, and this is the dark reaction. And the dark reaction starts with the fixation of the CO2 that you can see over here. So basically it has 3 stages. Possibly it will come later down in the question and we will see that, and it results in the production of the glucose. So number 1 is light reaction, and it is light dependent, so that's quite obvious answer over here. We can see A is the answer. Describe using visual the details of the light dependent reaction of the photosynthesis in eukaryotes, and that results in the production of ATP and NADPH. These are the final products. During photosynthesis, an electron transport chain is used to transport what? Of course, if it is an electron transport chain, so if it is transporting electrons, this is our one concept that we have to see. Now, how the electrons are being transported, you should know that the PS2 is the first one that transport the electrons, and here in between it is ETC, and then we have the PS1, and then it goes to the higher energy level, and here we have the production of NADPH, and here we have the production of ATP. So now you can easily see like what's happening over here. So ETC is connecting PS1 with PS2, PS2 with PS1, and the electrons are being easily transferred from PS2 to PS1. So now you can choose the answer for this question. So electrons from PS2 to PS1. Which of the following best describes the role of the water? Again, it's coming again and again. Above, we learned that the O2 is released, okay, and we also saw that over here from water the O2 is released. Now we need to see it in a different way. Water is the only source of the protons. No, we know that like water is the source of electrons, so we will go down. Water molecule donates electrons to the electron transport chain via PS2. Yes, we know that in this PS2, the photolysis of water takes place, like the splitting. H2O is resulting into half O2 each molecule of this one, and along with that protons and electrons are released over here. So B is the answer. We can see down why the C and D are wrong. Water molecules combine with the stored carbon molecule to produce glucose. No, we learned above CO2 is the one that converts into glucose. And water is the terminal electron acceptor. No, we know that terminal electron acceptor over here is NADP. So there is NADP plus that converts into NADPH. So that's the terminal electron acceptor. Question number 6. Which of the following best describe the relationship between PS1 and PS2 and the electron transport chain? Of course, all of them are responsible for the absorption of light, and they are working in a very sequential way. For example, PS2 comes first, then to electron transport chain, then comes the PS1. So all of these are working in a very sequential way. They cannot move here and there. PS2 comes first, of course, and that will be always the case. So they operate in sequence, with PS2 supplying electrons to PS1. Which of the following statement is or are correct about the photosystem? So photosystem, we know that like PS2 and PS1, these are the places where the light is absorbed. We know that light absorption takes place over here because they contain pigment, that's the chlorophyll, and also they have proteins. And since we said chlorophyll is involved in light absorption, so it means that they have the pigments also, like chlorophyll A, chlorophyll B, and all that. So in this case, it has chlorophyll center, yes. It has proteins, yes. It has complex of pigments, yes. So it means all of the above. Next question. Chloridazone is a herbicide that can be used to prevent the growth of many types of the weeds. A scientist wanted to determine how chloridazone prevents weed growth. The scientist grew seedlings of weed called black nightshade. The scientists applied chloridazone to half of the seedlings and left the other half untreated. So it means that he treated half with the chloridazone while the other no. So that you can see over here down, no chloridazone and chloridazone applied. Then what did he do further? The scientist measures the rate of O2, that's the oxygen produced in the abundance of NADPH, and the abundance of NADPH in the chloroplasts that were isolated from the black nightshade leaves. Scientists repeated the experiments both with the chloroplast exposed to light and with the chloroplast kept in the dark. So this is our independent variable now. The scientists obtained the data that is shown in the figures below that we have seen over here, but we will look into it. That's in the light and that's in the dark. While the other factors stay same, you can apply no chloridazone over here. So this is with no chloridation, and this one is with chloridation. So this is what you have to know. And also this is applied over here in this as a y-axis over here. This is the same thing. So percentage of the maximum level. Use the description of the experimental results. Okay, what the dotted lines show you, this is the O2 production, and what does the NADPH shows you, this is the abundance of the straight line shows you the straight line shows you continuous line shows you the NADPH abundance. Identify. So when they say identify, so simply identify, no need to describe or explain over here. The treatment or treatments, be very careful, that has or have the lowest level of NADPH at 12 hours. So we will see at 12 hours. So okay, it's here very high, 12 hours is low, 12 hours is low, and 12 hours is low. So we need to see has the lowest level. You can't see the lowest levels because it's same in all these three, so you can't see clearly which one is the lowest. So you can go with the answer mentioning all of these. So you can say like the experiment that's done in light when the chloridazone applied, the experiment done in the dark with no chloridazone, and experiment done in the dark with chloridazone. So three experiments over here that have. So you can say 3 treatments over here or 3 experiments over here. Identify the source of O2 that is produced in both treatments in the presence of light. So where is the presence of light? So this is the presence of light. We have a very high level of oxygen over here, and that is, we know from the previous multiple choice questions that the source is H2O. You can simply say H2O or you can say water. Based on the data provided, identify the photosynthetic process that is probably disrupted by the chloridazone. So photosynthetic process, we learned 2 processes above. If you go back here, so we learned 2 processes or 2 stages over here, light dependent or light independent. So it's quite straight away you need to say in this question whether if it is light dependent or light independent. And we can see over here in the dark there is nothing happening, like there is no change, no oxygen produced over here when the but over here when you applied chloridazone, the oxygen level is still there, but the NADPH has been used. It means that NADPH is not produced. And who is producing NADPH? Of course, NADPH is the product of the light reaction. So it means that which process has been interrupted by this one, disrupted by chloridazone. So of course you can say this is light reaction. You can use alternative names if you, for example, light dependent reaction, or we learned about as a Z scheme. So you can say that, or you can say photophosphorylation, but make sure you say linear photophosphorylation because we are talking about the light and NADPH source. Based on the data provided, explain why NADPH levels decline after chloridazone applied. So why it declined? Just now we said in the previous question, C part, we said light reaction is interrupted. When the light reaction is interrupted, it means that no more NADPH is produced. So all the NADPH which was present before the application of the chloridazone, that is being used by the second stage, that's the dark reaction. So all the NADPH has been used by the Calvin cycle or the dark reaction. Contrast, one more thing that you can see over here, always remember, like the things that you should know, the independent variable, dependent variable, and control variables. So you should know about these. In this case, independent variable, and that's always coming on the x-axis, right? But over here, x-axis, we have time is basically not an independent variable here. When I was reading up, I told you here these are the independent variables. So like if it is light availability, so independent is light availability, and dependent variable, of course, is always the y-axis, so that's the dependent variable. What are the control variables and what they have used over here? Like control variables are how much chloridazone has been applied, mean 1 ml, 2 ml, 5 ml, what type of the plant has been used, like the plant type, the temperature, the time. So you can say these are the control variables. Contrast cyclic electron flow and linear electron flow of photosynthesis. So it's quite obvious over here. When you move, I will highlight it with green. When you move from PS2, you go to electron transport chain, you go to PS1, you reach the final electron acceptor, that is NADP, and you produce NADPH. This is called as a linear electron flow. So this one that I have highlighted, it is the linear electron flow. While what is non-linear, that will be called as the cyclic. That is when the electrons flow only between the electron transport chain and the PS1. So this becomes the cyclic electron flow, and in the cyclic electron flow, the product is only ATP. No NADPH is produced. This is the difference between the cyclic and non-cyclic. And also you can see many more. For example, the linear involves both PS2 and PS1. Both systems are involved, while only PS1 is involved in the cyclic one. NADP and NADPH and ATP are the products of the non-cyclic, which is also known as the Z scheme, or you can say linear electron flow, while the only ATP is produced in the cyclic one. Which of the following is the process through which ATP is made as shown in the diagram? So we can see over here, this is the place where ATP is made. So of course ATP is produced from ADP. And how this is happening? This is happening by the addition of an inorganic phosphate. And the addition of the phosphate is called as phosphorylation. So basically, based on that, now phosphorylation is here, phosphorylation is here, and phosphorylation is here. So of course D is not the answer. So it is either A, but A says dephosphorylation, mean removal of the phosphate. No. C says photophosphorylation. Yes, that is photosynthesis. While the substrate level phosphorylation, basically this one we studied it in the cellular respiration process, that is the glycolysis and the pyruvate oxidation and Krebs cycle. So this is cellular respiration, that's the substrate level phosphorylation. A scientist is studying two types of the energy flow in the photosynthesis, cyclic and linear. If the scientists want to investigate a pathway that produces ATP but not ADP, H or oxygen, we just learned this by this cycle over here. So of course this is the cyclic one. Okay, so which pathway should they focus on and why? So linear, no. Linear, no. So either it is B or D. Cyclic flow because it generates ATP, yes. Without producing an ADPH, yes. Or oxygen, yes. Ensuring energy balance in the chloroplast, that's what you have to see. Yes, that's perfectly matched. But we will also read D, cyclic electron flow because it involves the splitting of water. No, splitting of water takes place in the PS2. That's not the part of this red circle, so that's the wrong part over here. Now here, the same question, you have to compare the pathways like the linear electron flow and the cyclic electron flow. So we have to see like what's the pathway of electrons. So of course, this is linear, we know that, and this is cyclic. Conditions under which the process occurs, like what type of conditions you need when this should happen. In the linear one, when you need ATP, when you need NADPH, and when you need oxygen, or you can just simply say NADPH and ATP, that's more than enough. And over here, when you only need ATP. Source, or you can say also like when NADPH is not consumed. Sources of electrons, who gives the electron over here? For sure we know that it is water, and over here it is PS1 itself. Electron acceptor, who is the electron acceptor over here in the linear pathway? That is the NADP plus. We learned it up over here. NADP plus is the final electron acceptor. And who is the source electron acceptor over here? Of course it will be PS1 itself. Who are the products? The products are ATP, NADPH, and O2, while only ATP is the product of the cyclic phosphorylation. Identify using visuals the three phases of the cycle. So you should know the three phases of the cycle, but memorization of the steps in the Calvin cycle and structures of the molecules and the name of the enzymes is not required. Be sure it's not required. So which of the following are the products of the light dependent reaction? So we just learned over here. This is also called as, both are basically called as light dependent reaction, but the linear is the complete one. The linear is the complete one, so light dependent. So what are, we learned up, we just wrote over here ATP, NADPH, and O2. List the names of the three phases of the Calvin cycle. I told you one phase, that was carbon fixation. You can say carbon dioxide fixation, carbon fixation, or only fixation. So whatever you want to say, you can say that. Number 2, it was reduction. And number 3, it is regeneration. Regeneration of who? Regeneration of Rubisco and RUBP. Okay, so that's the final product of the Calvin cycle. Explain using graph how, of course they say using graph, so you will be getting some graph. What are the factors that can affect the rate of photosynthesis? You should know that it can be CO2, it can be light, and it can be temperature, and all these factors are called as limiting factor. Okay, scientists examined the effect of the water levels on the net rate of the carbon dioxide fixation in the two types of plants. The plants were grown in either well watered soil or dry soil exposed to the similar conditions, concentration of the CO2. The net rate of the, so over here CO2 is acting as control. The net rate of the CO2 fixation for both the plants was measured. This is your dependent variable. Okay, of course whatever you are measuring, that's your dependent variable, and the independent variable is, of course, you are taking which type of the soil. So you need to use this to answer 14 till 16. Identify the name of the stage of the photosynthesis that involves carbon fixation. So of course we know that who involves carbon fixation, it's Calvin cycle. And identify the location. We learned it already. This is in the stroma. You learned it already here in this diagram. So be sure, like when you are working, you should know like what's going on. So this is the dark reaction. As I told you, dark reaction is over here, and that takes place in the stroma. We wrote it already. So you just simply say Calvin cycle, dark reaction, and it takes place in the stroma. Write one conclusion of the results of this experiment. So what's the conclusion of the results of this experiment? You can simply over say, like, when you use the dry soil, the net rate of the CO2 fixation decreases. So this is you can say as a conclusion over here. Explain the results of these experiments. Now here it was one conclusion, but here you need to explain, like, for example, the plants who are either in wet soil or in the dry soil, choose one. So you have to be very careful what you are choosing. Don't mix up all, because we need to explain the results. So you can simply say the plants who are in the dry soil did not have a water supply, and H2O could not split up, and since NADPH and ATP, these are the products of the light reaction which are required for Calvin cycle to produce to fix the carbon dioxide and produce glucose. That's not happening. So you can say they are not available to power the Calvin cycle. So with this we reach to end of 4.3. Since you can see over here it is 4.4. So let's pause this video here and we will go to part 2 for the respiration.